Hey guys, Michael and I are presenting on the paper by Irwin and Brody, published in 1999, called Nectar-Robbing Bumblebees Reduce the Fitness of Ipomopsis Aggregata Polymonaceae. I'll start out by saying that nectar robbing changes the behavior of pollinators. Nectar robbing can decrease plant fitness by damaging reproductive parts and deterring legitimate pollinators indirectly and directly. They can also enhance plant fitness by functioning as legitimate pollinators or accidental pollinators, and they help reduce gynoogamy, which therefore increases genetic diversity. As you can see in the diagram above that we modified from Professor Aiken's presentation, bumblebees have a direct negative impact on flowers they rob nectar from. Hummingbirds, on the other hand, have a mutualistic relationship with flowers because while feeding they help pollinate. Because bumblebees negatively affect flowers, they have an indirect negative effect on hummingbirds, which are true pollinators. The objective of this study was to determine the effects of nectar robbing by Bombus occidentalis on both male and female fitness components of the hummingbird pollinated plant Ipomopsis aggregata. The questions they were aiming to answer were: does nectar robbing affect the male fitness of I. aggregata on a per plant or per flower basis? And does nectar robbing affect the female fitness of I. aggregata on a per plant or per flower basis? The study was conducted in meadows near the Rocky Mountain Biological Laboratory in Colorado. The plant they chose to use is Scarlet Gilia. It's hermaphroditic but self-incompatible, meaning each flower has both male and female reproductive parts, but it rejects its own pollen and pollen of related individuals. The flowers of Scarlet Gilia are primarily pollinated by broadtail and rufous hummingbirds. This is done by the hummingbirds rubbing against the stamens and getting covered in pollen. When they move to another flower to feed, some of the pollen can rub off onto this new plant stigma. This is called cross-pollination. They are also monocarpic, meaning it only flowers once, sets its seeds, and then dies. The hummingbirds used in this study were the broadtail and rufous hummingbirds. Hummingbirds drink nectar by using their long tongues to gather the nectar from flowers. In doing so, they become great pollinators and form a mutual relationship with the plants they feed from. Hummingbirds avoid plants with high robbing and visit fewer flowers on those plants. The bee used in this study was the Bombus occidentalis. Bumblebees extract nectar using their sharp-toothed mandibles to chew holes through the side of the corolla, or petal, and inserts its proboscis into the hole, removing all available nectar without damaging the plant's reproductive or nectar-producing structures. They also do not pollinate the flowers and may reduce reproductive success through interactions with true pollinators. They examine the effects of natural and artificial nectar robbing on both male and female fitness components during the summers of 1995 and 1996. To assess the effects of nectar robbing on male fitness, they tracked dye particles from artificially and naturally robbed plants and estimated pollen, or dye, donation to other plants and local populations. To assess the effects of nectar robbing on female fitness, they measured it in 4 ways. They quantified pollen receipt using natural pollen loads, and subsequent fruit and seed production and seed mass from both naturally and artificially robbed plants. They verified that artificial robbing adequately mimicked natural robbing and its effects on both male and female function by comparing results from naturally and experimentally robbed plants for a week. They assessed the effects of nectar robbing on dye donation and pollen receipt in 6 populations, 3 in 1995 and 3 in 1996, and the effects of nectar robbing on fruit and seed production and seed mass in an additional 14 populations, 6 in 1995 and 8 in 1996. Different populations of Scarlet Gilia were used in each of the 2 years of study, and each population consisted of 200 flowering plants, totaling up to 4,000 plants. The methods they used to measure the effects of nectar robbing on female fitness for pollen receipt were by collecting all of the stigmas from focal plants, counting the number of pollen grains on each stigma, and estimating pollen receipt as the average number of pollen grains received per focal plant per week. They collected fruits from the focal plants in each population and recorded the number of fruits that did and did not set seed to measure the fruit and seed production. This was done in 1995 only. To analyze this data, pollen receipt was normalized with a square root transformation. Fruit set and seed per fruit were normalized using the arcsine square root and natural log transformations, and a mixed model repeated measures de novo was used to examine the effects of robbing on pollen receipt per plant, as well as fruit and seed production and seed mass per plant. To analyze the effects of nectar robbing on male fitness, they selected 15 random plants after dye application and collected the stigmas from half of their pistillate phase flowers. They quantified the amount of dye donated to outer plants by counting the number of dye particles on each color on each stigma. They analyzed this data by averaging dye particles per plant and dividing it by the number of plants dyed. They also normalized the data using square root transformation and mixed modeled repeated measures ANCOVA.
Some other methods they also did was they compared artificially and naturally robbed flowers by looking at 3 random plants per site and breaking the sites into high and low robbing and comparing them using statistical testing. And they also tested the nectar robbing on individual flower success, and they stole nectar from half the plants and colored the other half to exclude bees, and then assigned 2 different colors, and once again collected that data and then ran statistical testing. The effects of nectar robbing on male fitness was that male plants in high robbing treatments donated significantly fewer dyed particles than to plants with low robbing treatments, and it was significant. The graph shows all that. The effects of nectar robbing on female fitness was just as high. Plants in the high robbing treatment received significantly lower or fewer pollen grains than plants in the low robbing treatments, but that was only true in 1996 and not in 1995. For figures 3 and 4, fruit sets and seed mass did not have graphs, but fruit set within sites over both years of the study, the plants in, once again, high robbing treatments produced significantly fewer seeds per fruit than seeds from low robbing treatments. And then seed mass was also not concluded to be significant. Seed per fruit was significantly lower in plants that received high robbing treatment, as seen in figure 3, and plants in high robbing treatment produced significantly fewer seeds per plant than plants in low robbing treatments, as seen in figure 4.
All right, and that is all that we have for you guys. We will see you in discussion. Thanks.